Malaria. Malaria is a mosquito-borne infectious disease caused by protists of the genus Plasmodium. 4 species of the protozoal genus Plasmodium that cause malaria are Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, and Plasmodium ovale. Malaria is mainly transmitted through the bite of a female Anopheles mosquito. A brief overview of malarial parasite life cycle. When an infected Anopheles mosquito bites a person during a blood meal, it injects Plasmodium sporozoites into the subcutaneous tissue and bloodstream of a human host. Then, these sporozoites travel and enter the host liver cells, where they rapidly divide asexually. This asexual division, which is known as schizogeny, generates merozoites. The released merozoites invade other liver cells and enter the host bloodstream, where they invade erythrocytes. The merozoites divide asexually inside the erythrocytes. The erythrocytes rupture and a large amount of merozoites break out, bringing about the well-known cycle of periodic chills and fever, which is the characteristic of malaria. Some of the merozoites form gametocytes. When another Anopheles mosquito bites the infected host, it picks up Plasmodium gametocytes along with the blood. Inside the mosquito, male and female gametes form from gametocytes. Fertilization occurs in the mosquito's digestive tract and a zygote forms. An oocyst develops from the zygote in the wall of the mosquito's gut. The oocyst releases thousands of sporozoites, which migrate to mosquito's salivary gland, and from there, the sporozoites are injected by the mosquito into the bloodstream of a human host, thus starting the life cycle of the parasite again. Classification of anti-malarial drugs. There are various drugs that have been developed around the world in order to treat malaria. These drugs can be classified into several groups, which is quinolines, folate synthesis inhibitors, folate utilization inhibitors, and others. Examples of drugs under quinoline group are chloroquine, primaquine, quinine, and mefloquine. Under folate synthesis inhibitors, there are sulfadoxine and dapsone, while under folate utilization inhibitors, there are pyrimethamine and proguanil. Other drugs include helofentrin, atovaquone, and artemisinin. These drugs can also be classified into drugs acting on gametocytic form, erythrocytic form, and exoerythrocytic form. Example of drugs which acts against gametocytic and exoerythrocytic form is primaquine. Drugs which acts against erythrocytic form are chloroquine, mefloquine, quinine, and pyrimethamine. Among all, chloroquine is the most widely used anti-malarial and is the drug of choice in the treatment of malaria. Mechanism of action of chloroquine and other quinoline drugs. Chloroquine's mode of action is associated with heme detoxification. Ordinarily, parasites will digest the host cell's hemoglobin in its acidic food vacuole to obtain essential amino acids. This process releases huge amount of heme, which is toxic to the parasite. The parasite changes the heme to hemozoin, which is non-toxic to itself. When chloroquine is administered, it is concentrated in the parasite's acidic food vacuole, primarily by ion trapping. Chloroquine acts by binding specifically to heme, preventing it from polymerizing to hemozoin. This results in the accumulation of heme in the parasite and kills the parasite via oxidative damage to its membranes. Chloroquine resistance. Over the years, the emergence of chloroquine-resistant parasitic strains is rapidly decreasing its effectiveness. Chloroquine resistance is associated with a decrease in the amount of chloroquine that accumulates in the food vacuole due to an increase in the drug efflux. The increase in the drug efflux is caused by the mutation at the parasite Plasmodium falciparum chloroquine resistance transporter gene, which makes the Plasmodium falciparum chloroquine resistant transporter to efflux chloroquine at higher rate than non-mutated parasites. Chloroquine and its pharmacokinetic. Chloroquine is effective against erythrocytic form of Plasmodium vivax, Plasmodium ovale, Plasmodium malaria, and chloroquine-sensitive strains of Plasmodium falciparum. Besides, it also shows activity against gametocytes, except the gametocytes of Plasmodium falciparum. Chloroquine, however, does not show activity against the liver stage of the parasite. Chloroquine is administered orally, subcutaneously, intramuscularly, and through the rectal. 90% of the drug is absorbed from the gastrointestinal tract. It has a large volume of distribution due to its extensive sequestration in tissues of liver, spleen, kidney, and lung. More than half of the drug is excreted unchanged by the kidney, and the remaining is converted to active metabolites in the liver. Chloroquine is relatively safer anti-malarial. At therapeutic doses, it may cause dizziness, headache, diplopia, nausea, malaise, and pruritus of palms, soles, and scalp. Chloroquine should be used with caution in patients with hepatic disease, severe gastrointestinal, neurological, or blood disorders. The drug should be discontinued in the event of such problems during therapy. Today, malaria affects millions of people around the world, particularly in tropical regions, such as Southeast Asia, Africa, and South America. According to the statistics released by the World Health Organization, more than 200 million cases of malaria on an average are reported on an annual basis. It is hoped that with the ongoing efforts made by various organizations in the field of research, prevention and treatment of malaria, we could eradicate this disease and save millions of lives worldwide.